Good afternoon. I'm Doug Sesserman, privileged to serve as the CEO of American Associates Ben-Gurion University of the Negev, and welcome to #Webinar Wednesdays, our live from Israel Ben-Gurion briefings. It's noon Eastern time. I'm in my virtual office in New York City, and today we're joined by Professor Amir Sagi from Israel, where it's 6 p.m. This is going to be a fascinating talk about applied science and bioengineering at its best. The great mind, Amir's great mind, and the important work that he's doing at Ben-Gurion University. We're going to be talking about gender-bending prawns, and they're key to both disease elimination and helping the world's food security problems. Welcome, Amir.
I'm happy to be with you.
Okay, terrific. For those of you who have joined us before, you know the drill. If you have a question during the presentation, just click on the Q&A function. I'll be moderating those questions and we'll feed them to Amir. Amir has a presentation that'll last about 20 minutes. We'll use the final part of the webinar to just open it up to any question from the audience. And our goal, once again, is that if you give us 30 minutes, we'll give you a glimpse into one of the world's most exciting, dynamic universities and one of the most important institutions to the future of Israel. So our guest today, Professor Amir Sagi from the Department of Life Sciences and the National Institute of Biotechnology in the Negev. His influential research has significantly contributed to advances in the production of aquatic crustaceans as a protein source in many countries around the world, as well as scientific breakthroughs for disease elimination. He was one of the first scientists to demonstrate the significance of crustacean intersexuality, can't wait to hear about that, based on genetics and glandular function. His findings have led to greater understanding of sexual development in other diverse animal groups, can't wait to hear about that. He's also the co-founder of Enzutic, an agrobiotech company that has developed and commercialized a non-genetically engineered gender-bending treatment that allows freshwater hatcheries to produce crops of all female prawns. Professor Sagi will explain why there is a need for what we call these female super shrimp and when all male populations are a better choice. He's a brilliant man. He was a University of California, Hebrew University Reciprocity Scholarship recipient and a Fulbright Israel Scholar. He did his bachelor's degree in agriculture and animal science at the Hebrew University of Jerusalem, as well as his master's from Hebrew U in oceanography and marine biology, and received his PhD from the Hebrew University. Welcome, Amir.
Thank you. Could I share the screen now and start?
Yeah, start your presentation.
Okay, here it is. So I'm going to talk about biotechnologies for monosex aquaculture. I'll start with some basic research, some biology, and then refer to companies, products, health, and so forth. And so beginning is to understand where are we. Aquaculture, from the 70s, there is more and more aquaculture and less capture fisheries. Less fish in the ocean, more grown by men. And by 2050, we are going to see that majority of the food is coming from aquaculture.
Just looking at this slide real quick, Amir, so on a today basis, about half the fish is wild and half the fish is in hatcheries?
A little more is already from culture.
Thank you.
Okay, so we have the crustacean aquaculture, 2.6 million tons It's $69.3 billion. And we could see that it is really growing. There are several organisms that people are growing. And we will mostly talk about the giant river prawn, Macrobrachium rosenbergi, giant river prawn that we are working on in my laboratory. Look at the growth of this production and value. It's unprecedented growth of 22.3 increase since 2016 and still growing. So we are joining a very strong market. And just to understand, if we compare it, crustacean market by value and what we all refer to as the largest market in fish, okay? And you see the difference. So crustaceans are much higher in value, and lower in volume. And we are going to talk now about the size variation in prawns. And what we see here are females that are relatively uniform and males in which you have small ones, very large dominant males, and intermediates. So we have those two situations in the population. And if we look at the population as a whole, we could see that the females are around the 40-gram distribution. Then there are the small males, the intermediate orange claw males, and the large, between 60 to 100-gram large prawns. And the questions were, how could we help the growers around the world grow exactly what they want? Let's say that they want to grow the large sized. If we look at the large size, we see that those are only males. So how could we solve this problem? And this was initiated by our research at Ben-Gurion University, in which we discovered an insulin-like, hormone. And this insulin-like hormone that you see it here, which is quite similar to, if you look at here, the Homo sapiens insulin that we know of, responsible for our blood glucose, diabetics, and so on. But here we found that this insulin is responsible for maintenance in the creatures that we are working on. And that
do crustaceans actually have a pancreas like a human does that produces insulin?
They have something similar, but this insulin is not produced there. It is produced in an androgenic gland, which is responsible for maleness. That was a big surprise. And actually, if we look at it, it's like a switch. When the prawn is born, then there is a WZZZ, like we have the X, Y, and XX, okay? And the androgenic gland is formed, and then we have a switch. If we have this insulin, then it's on, and it's a male. Maleness is developed. And we have a male, a ZZ male. If
the shrimps, when they're born, are gender neutral, and then this switch makes them a male or female?
It's not exactly that they are gender neutral. They have their either WZ for a female, but ZZ for a male. But also us, we are at this point of the embryonic development. We are plastic. We could become a male or a female, okay? And if this switch is off, there is no male insulin hormone, it's a female. And this was the discovery. It's very simple. And now, what are you doing with such a switch? Okay, the IAG-switch. So we wanted to manipulate the IAG-switch. So we have, and here it's a little bit of biology. Okay, it's not going to be too heavy, but a little bit of biology we need. We have the DNA in every cells, in every cell of our body. And in the shrimp, there is a piece of the DNA that is transcribing for RNA that will be translated into the male hormone, this insulin, okay? So what were we doing in order to play with the switch? What we were doing what we call a temporal silencing of the gene without changing anything in the animal. We are just reducing the amount of the RNA, and if there is no RNA, there is no hormone. What happened to the genome of the animal? Nothing. So the animal is not genetically engineered. The animal is normal, and it's acting as a normal animal. It's not a GMO, it's a temporal, what we call RNAi or gene silencing in aquaculture. This was the first application of such a technology in the entire field of aquaculture.
Amazing.
So we started to do it in Vietnam with the growers in the Mekong Delta. And if you look at it, what happened, we have a female, which is WZ, and a male who is a ZZ. And if they are mating, we have 50% male and 50% females, okay? And now comes our trick. We are doing this silencing, the temporal silencing on a male, and this male becomes a female. But it is not changing its genome. This female is a ZZ female, okay? So if we cross it with a normal male, the resulting progeny will be 100% males. And this is exactly the technology that was patented by BGU. And now they're selling more over, it's the second year or third year that they're selling. Only in Vietnam, they're selling over $1 million worth of 100% ZZ post-larvae. So, and this was made in collaboration with a company from Israel that got this exclusive license on our patent. So now I'm going to the second part of my talk, and this is about the all-female. So what happened if a commercial grower doesn't want to grow these large animals that are scarce in the population? He wants
Amir,
to grow.
do they, I know these shrimps are as close to being kosher as a shrimp could be, but how do they taste, these larger shrimp? Do they taste the same as the smaller shrimp? They're just larger in size.
As far as I know, there's no difference in taste. The difference is in size and capacity of yield. So if you want to, if you don't go to the premium size, but you go for a higher yield of medium size, then you want to have only the females, okay? And then you will have a very elegant crop that is very uniform. So that was another challenge. And then we had to go for that, and we established with the university a startup company, and you see the founders by the name of Enzutik. And this company is situated in Beersheba and working on shrimp all female, is doing a different thing. If we're working on a female and a male. As I said, ZZ is a male and ZW is a female. We cross them normally, we will get a 50% female and 50% males. Now comes the issue of Enzutic. What Enzutic was doing, it was creating something that is not there in nature. Again, no genetical modification, a super female, which is WW, okay? And then if you cross this super female with any male, it will be all female. So that's the technology of Enzutic. And if you can see a mixed population, you remember small males, females, medium size males, large males, a lot of differences in the population. But now, if you grow all female, look at the product. It is very uniform, okay? One major problem is those snails, and this is something that we took a picture in the northern part of Israel. And those snails are very abundant. Some of those are invasive species, very much abundant in fish ponds, and they're carrying parasites that are affecting the fish that they're growing in the pond. And here is what we are doing. We are putting we are using the monosex prawns as biocontrol agents. And here, this is a pond up north that in which we stocked biocontrol prawns. And here is a pond that we didn't, okay? And there's a big difference in the abundance of the parasite-carrying prawns. So why do we need all?
So the snails carry the parasite or the infection, and the prawns are predators for the snails?
The prawns are predators of the snails, and only when you have a solution of a monosex that is not able to reproduce in the pond, then you could use it because it's safe and not producing invasive species in the areas. So, and here you can see that when you have the snails, you have parasites in the gills of the fish, and when you don't have, the gills are clear. And now I'll end my talk with another parasite, very similar to the parasite that we saw in the gills of the fish, but now in human beings, and this is bilharzia or schistosomiasis, in which more than 250 million people are required preventative chemotherapy against this disease in Africa mostly. It's caused by the same mechanism of a parasitic worm that is growing in snails and then infecting a vertebrate. That's the life cycle. We have a human being and we have a snail, and the parasite is transmitted from the snail to the human being. That's a very devastating disease, and it's a disease of poverty. What are we suggesting here is to use our monosex prawns in Africa, and this week we had the first harvest of all male prawns in Senegal. The first harvest ever of all male prawns in Senegal, and now the Senegalese authorities have approved future introduction of monosex prawns into bodies of water like that to reduce the amount of schistosomiasis or bilharzia carrying worms and protect the health of people that are coming here to this water to use the water, as you can see, and to wash their clothing and to
So humans can get this disease just from the water itself, by drinking it or washing their clothes, not from eating the snail?
No. They're getting it only when they are inside the water, and then the parasite is it's like a torpedo jumping from the snail through the skin of the human being and penetrating the body. You cannot get it from the water that you're taking. You have to be inside the water to get it. So the idea is...
And this is the project that was funded by the Bill and Melinda Gates Foundation, correct?
Exactly. And now we are at a different step there. That
Well,
we want.
One last question. You mentioned that these are monosex prawns. Can it be a monosex, either all female or all male, and have the same effect?
Exactly. Monosex is for either all male or all female. At any rate, a population that does not have both sexes and cannot reproduce in an area that it could become an invasive species. Yeah, here you see Bill and Melinda Gates that were funding us, other than many other sources. Okay, so up to this point, and I'm opening this for questions if you have.
Okay, so let's stop the share. And to our participants in the audience, to ask a question, you just click on the Q&A function, and I'll be able to read your question and get it to Amir. So while we're waiting for some questions, that was a fascinating presentation, really amazing. Real quickly, you're from outside of Jerusalem, you went to Hebrew U. How did you end up in the Negev? And tell us why Ben-Gurion University is the place for this outstanding research.
Yeah, always asking how come a marine biologist in the middle of the desert? So I started my studies at Hebrew U, and then I was during my PhD at University of California, Santa Barbara, and UC Davis at the Marine Station, the Bodega Marine Lab. And for postdoc, I went to Woods Hole, Cape Cod. And as a marine biologist, I always wanted to work with aquatic organisms. And then came this offer from Ben-Gurion University, and they said, yes, you can do it in greenhouses. And you can do it in close recirculated systems, which is actually, at that time, it was like, this is illogical. But right now, most of this industry is going into this direction. And at the end, it will be mostly close recirculated systems, very close to urban places that will be holding zero discharge and will have the product, let's say, the farm very close to the fork.
Okay, we're getting lots of questions. I'm going to try to feed you some of them. They're pouring in. Steven Sirkin is asking, how long do these monosex prawns live while they're in these ponds or lakes as predators for the parasite?
In the fish ponds, the systems are very strict. Fish ponds are functioning for 6 months, and then they're harvested. In the natural habitat, they could go as far as 2.5 years.
Wow. So Jan Liff one of the people very close to us and on our board, she's asking, some humans develop shellfish allergy from farmed shrimp. What chemical causes that?
There are several chemicals, and it's not from farmed shrimps. It's from shrimp. People are having some allergies, some that are related to certain proteins in the flesh of crustaceans, and it has nothing to do with the issue of either farmed or not farmed. It is a crustacean. It's a crustacean protein, and such people have to be very careful in dealing with any shrimp flesh. But as Jews, we have no problem like that.
All right, we have speaking of being Jewish, we've got a good question from Betty Ruth Walter. She's asking, how does one do research on a food source that isn't kosher in Israel? Who does the taste testing?
Okay, so it's a sensitive question. First of all our funding sources are outside of Israel, and our markets are totally outside of the country. We are producing the ideas. We are producing the biotechnologies. We are not producing any food item here. There is no culture of shrimp in Israel whatsoever. And this is not because of kosherness. This is because it's too expensive to grow crustaceans in Israel. Labor is too expensive. Water is all desalinated, very expensive. And space.
And my understanding is, all meat in Israel is kosher, but there are many non-kosher restaurants where you might find even shrimp on the menu.
Oh, yeah.
Okay. Sure. So Sister Abigail Judah, who's...
it's a liberal country.
Yes. Sister Abigail Judah, who's one of our regular webinar participants, she asks, is there a way for people to heat water before they bathe in it so the parasites don't enter their bodies?
As I said before, we are talking about a disease of poverty. And we are talking about people who are using the sources of the water. It's not a matter of treating the water. It's a matter of going into the sources. Each village has a small source of water, and the women are going there to do their laundry. So they are sitting inside. I saw some people coming with their cows. So they're going with the cows inside so they can drink, okay? They were washing their horses. They were doing things that require direct contact with water. And when you are in direct contact with the water and there is a parasite carrying snail around, then you might be infected. I never go to the water.
One last question from one of our anonymous attendees is asking, can the technology be used to produce insulin, the same thinking you have to dampen the gene, if you will? Can that be used to produce insulin?
I didn't get it. To produce insulin?
Yes.
We are doing exactly the opposite. We are reducing the expression of the gene that is translated to insulin. So we are reducing the amount of this particular insulin that is the male-specific insulin. We are reducing it in the body for a few days, and this is enough at that point for the animal to transform into a female.
Thank you very much. So this webinar was called for 30 minutes. Professor Sigi, you did a tremendous job. This is just fascinating research. I'm so pleased that Bill and Melinda Gates are helping solve this disease in Africa and other parts of the world. It's our pleasure to work with them. So we want to thank you very much for the work that you do and for joining us today. Our next hashtag webinar Wednesday, for those who want to join us, is on Monday, November 23rd, following Ben-Gurion Day. We're going to feature Dr. Paula Caballo and have an inside look into the Ben-Gurion archives. David Ben-Gurion, the founder and first prime minister of Israel, and the guy whose name is on the door of our university, once said that in the Negev, the Jewish scientific and research ability, as well as the creativity and pioneering valor of Israel, shall be tested. Amir, you represent all of those qualities and one amazing scientist. Thank you so much for your work. Thank you to all the participants for joining us. Be safe and stay healthy.